God's wonderful world. The Book of Genesis. The Book of Genesis chronicles the creation of the world, the universe, and the earth. It reveals the plan within God's heart to have a people of His very own, set apart to worship Him. The author of the Book of Genesis, Moses, is credited as the author. The date, approximately 1450 to 1410 BC, is written to the people of Israel. The landscape of the Book of Genesis. Genesis is set in the Middle East region. Places in Genesis include the Garden of Eden, the mountain of Ararat, Babel, Ur, Haran, Shishim, Hebron, Beersheba, Bethel, and Egypt. The theme in the Book of Genesis. Genesis is the book of beginnings. The word means origin or beginning. It sets the stage for the rest of the Bible, telling us God's plan for His creation. Genesis reveals the nature of God as creator and redeemer, the value of a human life created in God's image and for His purpose, the terrible consequences of disobedience and sin, separating man from God, and the wonderful promise of salvation and forgiveness through the coming Messiah. The Book of Genesis has several key characters: Adam and Eve, Noah, Abraham, Sarah, Isaac and Rebekah, Jacob and Joseph. In Genesis 1:27 it says, "So God created man in His own image. In the image of God He created him, male and female He created them." So God created life on the planet Earth. And now scientists have actually done research and tests, and they actually found our DNA. Our DNA. And today, brothers and sisters, that is what we're going to discuss. DNA, which is essential, vital to life. DNA, deoxyribonucleic acid, is a molecule that carries most of the genetic instructions used in the development, functioning, and reproduction of all known living organisms and many viruses. DNA is a nucleic acid alongside protein and carbohydrates. Nucleic acid composed the three major macromolecules essential for all known forms of life. Most DNA molecules consist of two biopolymer strands coiled around each other to form a double helix. The two DNA strands are known as polynucleotides, since they are composed of simpler units called nucleotides. Each nucleotide is composed of a nitrogen containing a nuclear base, either cytosine, guanine, adenine, or thymine. as well as a monosaccharide sugar called deoxyribose and a phosphate group. The nucleotides are joined to one another in a chain by covalent bonds between the sugar of one nucleotide and the phosphate of the next, resulting in an alternating sugar-phosphorus backbone. According to base pairing rules, hydrogen bonds bind the nitrogenous bases of the two separate polynucleotide strands to make double-stranded DNA. The total amount of related DNA base pair on Earth is estimated at 5.0 times 10 to the 37th power and weighs 50 billion tons. In comparison, the total mass of the biosphere has been estimated to be as much as 4 TICs. Okay, we discussed the chemical makeup of DNA. So, what is DNA? Where is it found? What makes it so special? How does it work? In this article, we will look deep into the structure of DNA and explain how it makes itself and how it determines all of your traits. First, let's look at how DNA was discovered. DNA is one of the class of molecules called nucleic acid. Nucleic acid was originally discovered in 1868 by Frederick Mitscher, a Swiss biologist, who isolated DNA from pus cells on bandages. Although Mitscher suspected that nucleic acid might contain genetic information, he could not confirm it. In 1943, Oswald Avery and colleagues at Rockefeller University showed that DNA taken from a bacterium, Streptococcus pneumonia, could make non-infectious bacteria become infectious. These results indicate that DNA was the information containing molecules in the cell. The information roles of DNA was further supported in 1952 when Alfred Hershey and Margaret Chase demonstrated that to make new viruses, a bacteriophage, a virus injected DNA, not protein, into the host cell is needed. So scientists had theorized about the informational role of DNA for a long time, but nobody knew how this information was encoded and transmitted. Many scientists guessed that the structure of the molecule was important to this process. In 1953, James D. Watson and Francis Crick discovered the structure of DNA at Cambridge University. The story was described in James Watson's book, The Double Helix, and brought to the scene in the movies. The race for the double helix. Basically, Watson and Crick used molecules, modeling techniques, and data from other investigators, including Maurice Wilkins, Rosalind Franklin, Erwin Chargaff, and Linus Pauling, to solve the structure of DNA. Watson, Crick, and Wilkins received the Nobel Prize in Medicine for the discovery of DNA structure. Franklin, who was Wilkins' collaborator and provided a key piece of data that revealed the structure to Watson and Crick, died before the prize was awarded. As you can see on the screen, replication prior to cell division. So before a cell can reproduce, it must first replicate or make a copy of its DNA. Where the DNA replication occurs depends upon whether the cell is a prokaryote or a eukaryote. Like the One Ring of Power in Tolkien's Lord of the Rings, deoxyribonucleic acid, DNA, is the master molecule of every cell. It contains vital information that gets passed on to each successive generation. It coordinates the making of itself as well as other molecules, so-called proteins. If it is charged slightly, serious consequences may result. If it is destroyed beyond repair, the cell dies. And we're talking about cells in our human body. The presence or absence of DNA evidence at a crime scene could mean the difference between a guilty verdict and an acquittal. DNA is so important that the United States government has spent an enormous amount of money to unravel the sequence of DNA in human genomes in hope of understanding and finding cures for many genetic diseases. Finally, from the DNA of one cell, we can clone an animal, a plant, or perhaps even a human being. So, my brothers and sisters, as you see, DNA is very, very important. How important is DNA? Well, each person has 23 pairs of chromosomes, one unique you. Okay? That's what makes each one of us unique. You are made up of cells, and the cells in your body has 23 pairs of chromosomes. Your chromosomes are made of DNA, which can tell you a lot about you. And this is how they determine your DNA makeup, to learn about your ancestors. So DNA analysis is performed in laboratories that are certified to meet the CLIA certified, which is Clinical Laboratory Improvement Amendment of 1988. That's what CLIA stands for. Okay? So again, even in science, they have standards. A CLIA certified lab must meet certain quality standards, including qualification for individuals who perform the test and other standards that ensure the accuracy and reliability of results. They are to use leading technology to genotype your DNA. Now, I mentioned about an agency called 23andMe. The 23andMe difference, and here's what you get: a genetic lab test or ancestry service. What they do is explore your genetics throughout your life, from a genetic snapshot of your health and ancestry to ongoing reports and research. Everyone learns something different. As the field of genetics advances, they will be there to make the latest technology accessible to you. So how does this work? It's just saliva. Provide your saliva sample from home. Mail it to the lab of your choice. in the same kit it came in, and the postage is prepaid. Then they bring your genetics to you. So you mail it in and they mail it back. So you may question, what is the history of the company? How accurate are the reports? How is your privacy protected? Now remember, 23andMe is just one of the agencies that do DNA testing. There's Ancestry.com and others. So this is the statement that 23andMe says. Our test can be used to determine carrier status in adults, but cannot determine if you have 2 copies of a genetic variant. Each test is most relevant for people of certain ethnicities. The tests are not intended to diagnose a disease or tell you anything about your risk for developing a disease in the future. On their own, carrier status tests are not intended to tell you anything about the health of your fetus or your newborn child's risk of developing a particular disease later in life. Okay, so that's the statement that 23andMe makes. So today, we went through DNA testing. And this right here, when you look at the diagram that I presented to you, it shows you that there has to be a higher power than man to create such a complex mechanism as DNA, which makes up the lives of every living being on the planet Earth. If it lives, it has a DNA. So God has to be real. Man could not do this on his own. Evolution could not create such complex people. Remember, brothers and sisters, God is all science. It took scientific knowledge to create man and all living things. Be blessed. 3 years ago, when we started these videos, we talked about the genetic testing. And so today, I'm happy to say, we could follow up on it. Father, thank you for allowing us to follow up on genetic testing. Bless your people and bless the peace in Jerusalem. Have a blessed day.